So you need to know tests in order to know how we can test for different types of food categories, including glucose, starch, protein, and fat. So we're just going to go through the different practical methods for testing for each of them, as well as the correct colours to show that a test is positive or negative. So the first one is glucose. We prepare a food sample and we put this in we transfer this in 5 centimetres cubed to a test tube. We prepare a hot water bath at 75 degrees roughly, and then we add some Benedict solution to the test tube with about 10 drops, quite a few, to the pipette. Then we place this test tube with the food sample in the water, leaving it for 5 minutes and making sure that the test tube is pointing away from you. Once we've done this, if it's in low concentration, then we'll get a light green colour. But if it's in a high concentration and the test is positive, you get a brick red colour. If the solution has not been tested correctly for glucose or it's negative, then it will be turquoise, showing there's a normal concentration of glucose. If we test for starch, again, we make a food sample and we transfer 5 centimetres cubed of this to a test tube. We add the iodine solution and we gently shake the test tube to mix all of this, and if it's positive it will turn navy, and if it's negative it will just stay the same colour. For proteins, again we make a food sample and we transfer 5 centimetres cubed to the test tube. This time we add a small amount of bioarray reagent, we swirl and we observe the colour after 5 minutes. A positive result in a high concentration is turquoise/pink, but if it's negative it stays the same colour. Lipids are quite easy as well. We prepare a food sample and we transfer 5 centimetres cubed to a test tube. We add a bit more than last time of ethanol, and the one thing we do differently than usual is we add a bung on top immediately. One of the ways to test this would be to get a red colour at the top of the tube for a positive result, or a negative result would be stays the same colour. However, you can also test and get a cloudy observation if it's positive. There are 2 ways to tell.